So is he. So hopefully you guys can come back in soon and that this stream works better. So I'll wait for a few more of you to show up. I feel like I'm making some sort of mad scientist face. Sorry. Today is still Monday. Molecular orbiting techniques. Oh no, I don't know. I'll give you guys another couple of seconds. Y'all, of all the days have technical difficulties. We're so close. I think there were more people watching, so we'll keep waiting, giving people just a second. Oh, good. I'm glad you can see us. I'm glad you are not watching orbiting techniques or something else. Hold on. Okay. So there are a few of you, hopefully more, we're hanging out. So today, well, I know that more people are coming, so I will wait. But also, I'm gonna go check on the dog who's making like a huge ruckus. Nope, never mind. I can see her. She is playing with toys. Crazy. Okay, looks like more of you are starting to trickle in. I think it sent many of you to the wrong place, and I'm really sorry. So hopefully it will all kind of come together. So just to recap where we were before we had massive technical difficulties of black screens. Oh. I don't really know a lot about how YouTube works, but mostly this kind of works. So we were discussing, well, I was discussing, I don't know who else was here. I'd like to apologize. I'm glad you guys have made it. Feels like the first day of school where we went to like 9 different places to try to learn. It's a hard life. So before the internet rudely interrupted our lives together, we were talking about restriction enzymes and sequencing. Likely you would do both. In today, I don't want to say today land. So currently in science, the price of sequencing per construct has decreased to where it's close to the restriction digest price. The advantage is that it can get expensive to do this on multiple constructs all at the same time. So usually you run a restriction digest and then you will go back and sequence later. This tells you, do you have the DNA construct that you intended to make? Knowing what you have helps you be able to address the question, what does this protein do? So when we think about what does this protein do, that's impossible to ask if there's something wrong with the protein we made. So from there, we can start to look at, I wonder if there's something. I'm gonna close a bunch of stuff on my computer. I don't know what that's gonna do, but like, I feel like I'm very close. Like if you came back in, like, I feel like I'm very much in your face. Yes, our New Year's resolution should have been Wi-Fi without buffering. I'm all on board with that. Oh, we're back. Basically, I'm trying to like close out anything that could be using the internet on my computer. I know, life is hard. And we will turn this off. Let's hope all that works. Because basically, I put my phone on airplane. I turned my other laptop because my work computer is behind it. It doesn't have internet. So really, there's like only internet going to this. So we'll see how it goes. Hopefully things are better now. If it's still all wonky, we will, I don't know what we're going to do, but we're going to do our best. Actually, yes, I do. I will restart the internet program and then we will try again. So hopefully it's good now. So what was I talking about? Oh, protein analysis in the cell, or it can mean in life. And so in this case, we want to ask questions like, how does it function in the cell? So depending on what you think the protein's job is, you could try to alter its behavior inside the cell. If the protein is an osmotic response protein, you can change the osmotic environment and see how the cell survives. If the protein is involved in amino acid synthesis, you could see if it was very promiscuous, right? So we know that most enzymes are not very promiscuous. In this case, we mean that they would accept more than one starting material. So you could try to alter that. You could do feeding studies. You could do lots of things. We could also purify the protein. We could do that using a his tag, or frankly, any other tag. So if we can purify the protein, we can ask about it in a really controlled environment. So if you guys remember back to Biochem 1 and Michaelis-Menten plots and Lion-Weaver-Burke plots, don't worry, that's not on the final. But those plots look at enzymes and they ask, okay, how does this enzyme behave? So we can do that with a purified protein. So here we can start to ask, with a purified protein, we can ask specific controlled experiments. So specific controlled experiments here can start to say things like, what does it do? If we add different amounts of reagent, do we see different behavior? So that works best if we have pure protein. So pure protein is going to behave. Lily, come here. Come on, honey. Hold. I'll be right back. Hey, come on. Come here. Come on. Hey. Well, come here. Come here. Hey, come here. Sorry. The dog really at 5:05 is like, I'm done with life. Do you want to say hello to my students? Mostly because it's Monday. Yeah. Here she is. Hey, say hello. I'm embarrassed for my dog's behavior. But everyone needs a little dog life. Back to real life. So when we think about this, one of the things with the specific controlled statements is that we can change the environment easily. We can easily change the, let's be seen, yeah, environment. So the advantages to changing the environment is we can ask, what happens if we change the salt? What happens if we change the buffers? If we add 2 competitive inhibitors to this enzyme? If we're thinking about a methyltransferase, so a methyltransferase does one thing. It takes a methyl group from object A and moves it to B. Well, what if we change the structure of A? What if we change the structure of B? So in that case, we can do all of that with pure protein. So for the next couple minutes, I want to think about what other types of protein analysis that we talked about earlier this semester that might be advantageous in this case. So if you vaguely remember, hopefully you do, it'll be on the final, unnatural amino acids. So unnatural amino acids are where we take these chemical components and use them in place of amino acid 1. So point mutations so we've talked about the ability to take, let's change glycine 105, and we want to make it into a tryptophan. So this is our starting amino acid. And we've created this. So we have a glycine, and we want to know if we take the smallest amino acid and make it into the largest amino acid, does the protein still function? So when we think about this, one of the points of this that is really important is we need to be able to compare. Yeah. Compare, in this case, we're going to say before or wild type to the after mutated. So one of the things that is really important to think about with this is if we change the glycine to a tryptophan, we need to know what we're asking. If it is an enzyme, we could ask, does the rate change? So if you vaguely remember Michaelis-Menten, well, not Michaelis-Menten, but a Lineweaver-Burke, maybe the slope of that line is going to change. Maybe it's going to move up and down on the graph. So that's one option. If you are looking at a methyltransferase, maybe it doesn't accept methyl groups anymore. Maybe instead of methyl groups, what about now if you change this, it will accept ethyl groups. So instead of a CH3, now we're going to add on an ethylene group. So point mutations, we can set this up. My group makes a lot of these. If we know what this amino acid does, what happens when we change it? And so if you remember this discussion with unnaturals, this does give us limited options, right? Limited options meaning we can only use the regular amino acids. But there is enough chemical variety for you to ask a lot of questions about your particular protein. So one thing you should kind of be able to see today is that the opportunities in molecular experiments are endless. So you could ask about a point mutation. You could use an unnatural amino acid in order to change, instead of glycine 150, we could do an MT. We could also use unnaturals, amino acids. We could use MT set, MTS, we'll say MTS reagents. So our goal in all of these is to be able to ascertain or determine, what does this protein do? And so by changing its structural shape or the binding pocket or something else, we can now begin to say, like, okay, if we know what it does, how does it do that? And so you can do that by looking at different things. So I also want to think about, so we know what it does, but what does it look like? So on the very first day of class, we talked about protein structure. And in that discussion, it said we needed to have pure protein. So purifying the protein is often just the first step. Purify the protein, and now you want to run experiments on it. So what about if we want to know, what does it look like? So protein structure is advantageous because it's going to tell us about the interactions that take place. So if we want to know what does it look like, that can tell us what point mutations to make or other types of information about it. So we could use X-ray, cryo-EM, how do I spell that word? We could use, there are some labeling techniques. We could use our, well, my best friend, Fred. So we could use FRET. Now, FRET is an interesting thing. It can tell us, do two proteins come together? It could ask us, does this protein arm do this? And we can track all that with changes in the transfer efficiency. So all of the different techniques we've kind of met along the road all build together to give us more information about the proteins we care about. So these tell us about the structure. And so we know that a high resolution structure tends to be on the order of 1 to 2.5 angstroms. So oftentimes, I know for myself, I have to kind of think through, like, is it that it is a high resolution, meaning we can see lots of information, 1 to 2.5 angstrom resolution, or when we say low resolution, that usually means that it is more of an overall picture, kind of a shape graph, to say like, hey, it kind of looks like it fits like this. So all of these can give you information about different regions. So the FRET and the labeling can tell you about movement. FRET would be really hard to use to determine the structure of a protein. You would need to know, like, hey, we think these two regions cross, or in that case. So these are different ways that we can start to think also to answer, what does it do? So the ability to discover what a single protein does can have far-reaching effects. So we can go from something similar to, what does this protein do? To, we purified it and now we want to know, we've made changes to it, what does it do? So this kind of information is good if you've discovered the protein and you want to make changes, or we've changed the protein that we already know a lot about and we want to know what happens. So sometimes proteins have regions, right? It can have other types of domains. So scientists like to know, what happens if I cut it off? If you insert a stop codon in the middle of a protein, can it still do its job? And so you have to have assays to do that. So the things that all of this molecular techniques has really come together, all together, it basically tells us, what do proteins do and how do they do this? And so for every protein, all of these experiments are different. So Ryan asks, is labeling the technique that was used in the Hershey chase? That is a labeling technique. So labeling can mean we used radioactive entities and then saw where they went. You could also do fluorescence labeling, where you stained for particular parts of a protein using small molecule fluorophores that bind specifically. You could, so there is another labeling technique, which I don't think we've talked about this semester. So if we have a membrane and you have a protein that looks like this. So this is outside of the cell, this is inside, and this is a membrane. If you were to add a small molecule, here's the blue one. You could add reagents that will basically bind out here and run on a gel. So if you had something here, say this is a his tag, you could ask questions. So if these blue dots were reagents where it would basically cut the protein in different places, you would be able to see a map of different lengths of proteins. So this is not helping you. If the label cleaves the backbone, you can map topography, that's a big word, on an SDS page. So basically, if in different regions you could cleave those, then you could set them up in that way. So when we think about labeling, you could do that. There are also labels that can transverse, so they can cross the inside of the cell. And then you could do similar experiments on the other side. So these experiments would likely have moved in the recent past from the state-of-the-art to classic techniques. Today, there are other ways that you could kind of predict this. But I don't want to say in the olden days, because like I've done these experiments in my career, not in a lab. One of the things that this is good for is if you don't know a lot about your protein. So if you're trying to ask how many transmembrane domains does it have? So you can label the outside of a protein versus the inside to basically see what is exposed and what's not exposed. And so there is also the labeling that you're talking about where you add in a radio label, which is different than this in this case. Good question. So other things that we can really think about in this molecular technique. So we've talked about RT-PCR, we talked about bioinformatics, we talked about FRET, we talked about structural proteins. So all of this is to say that the ability to ask questions about your protein, I don't want to say is endless, but there are lots of options. And so we can use all types of different techniques to basically try to give us insight into what these proteins do. And you can make changes to them, and you can do all of that using the techniques we've talked about. So point mutations are made using PCR. There are ways that you can force it to make mistakes. And when you force it to make mistakes, you get something new and different. Trying to think what else we could talk about. Well, I mean, I have lots of things. Trying to make sure we've covered all the points. Does anyone have any questions about molecular techniques or any of the techniques that we've talked about in this section? While I close the window. That's awkward. So on the final, I'm going to ask questions. Tell me about this technique, or look at this data and analyze it. So all of the things we've really talked about are giving us insight into how proteins function. And so what we want is for proteins, for their function to be something that we can ascertain and try to learn. So I guess I'll hang out here for another minute and a half or so. If you have any questions, you can drop them in the comment box. And I'll answer those. Otherwise, we're done for today. I think we've kind of reviewed from a much higher level about all of these techniques. And I will post the bioinformatics information from last year here in just a second that you can kind of give it a go. If you have questions about bioinformatics, I'm happy to help, or I'm happy to give you insight if you do that as part of your research. I can tell you that I miss you guys. Like a lot. Hopefully you guys are all doing well. If you have questions for the review, send me in the comments box. I was going to run it elsewhere. Previous take-homes required the internet. You will, so Ryan is asking, the previous take-homes required the internet. Will the internet be required? So yes, but you will be required to submit your take-home on the internet. Will you have to go to websites? Likely. I will ask you to go look information, go look at where this product comes from and tell me about it. If you don't use the internet. Hey, Ryan, it might be best if you pop into office hours or pop in, like, send me an email. It sounds like you're asking a really specific question, and I'm not entirely certain what it is. But you will need access to both the book and all of your notes. So you should be able to do most of it without that access. But because it is open internet, I likely just send me an email or pop in office hours and ask, because I think you're asking something specific, and I'm not sure I'm answering that. Any other questions today? Transformation. So Johnny asks, the transformation techniques are used to force DNA to diffuse into the cell. Where does the vector come into play for this process? In that process, the vector is the DNA that you're shoving across the membrane. So the goal is you make the vector basically in a test tube, and in order to use the DNA to study the protein, you have to do it inside the cell. So after the ligation reaction, you have a plasmid, just a piece of DNA. And in order to use that piece of DNA, it has to be housed in an expression system. There are some, full disclosure, in vitro expression systems where you basically add a bunch of stuff into a test tube. But even in that case, you would put it into a cell first. So the vector is the DNA that you put across. So the vector, which in that case is the plasmid plus the gene, has to go into the cell. So that is the DNA that we're talking about. Does that make sense? Sorry, life is hard. Any other questions? I wish I was getting to watch you guys walk across the stage next week. But we all know that. I'll give you guys about another minute or so if you have a question. Otherwise, my office hours start in about 5 minutes. I'll hop in there early in case you have a question. All right. Well, it doesn't look like there are any more. If you send me one, you can send me an email and I'll answer that. Y'all have a fantastic Monday. It looks like it's sunny here in Jacksonville. So enjoy.